basically. So I'm going to give you a couple of information about Claire before we start. Claire did her PhD and postdoc at the University of Sydney and the Australian Nuclear Science and Technology Organization. And their main focus was the study of membrane protein and small molecule binding, which I know it's an interest of many other people here at Essex. So I hope you will enjoy the talk. Specifically, she studied the assembly, so affinity measurement binding of lipid bilayer and protein lipid bilayer. And she also studied this system at a larger scale to determine both the structure and the composition. And she mostly used neutron and X-ray scattering. The main protein that she was focusing on was TSPO, which is a mitochondrial protein, actually a mitochondrial transmembrane protein. And this protein is quite interesting because it's known to have a role in neuroinflammation, and it's upregulated microglia during neuroinflammation. So after a PhD and postdoc, she, for some reason, she moved to UK and she joined as application specialist at NanoTemper, and she supports customers in UK and Ireland, like me. And so Claire, I'll leave it to you.
Okay, great. Thank you, Filippo. Yes, so my name is Claire Hatty, and I am now an application specialist at NanoTemper. But as Filippo said, I am originally from Australia, but I very much enjoy living in Cambridge. So today I'm going to talk to you about understanding biomolecular interactions throughout the drug discovery process with MST and TRIC. So I'm going to be talking about two main technologies in today's talk, which are MST and TRIC. I know it does say drug discovery in the title, so there'll be a little bit about drug discovery, but also it's just going to be about generally how we can use those technologies for studying any sorts of biomolecular interactions. So I've included a few examples that I think would be most relevant to you. So hopefully there's something in here for everyone. I aim for it to be no longer than 30 minutes. So just maybe bear with me through the bits that if you're not so interested in. So we're just going to basically briefly go through the background to the technology and then move on to some specific examples. So I just want to start by first, I guess, exploring the question of why we would want to measure biomolecular interactions. And generally it's because studying binding affinity enables us to better understand cellular processes and cellular signaling. So first we might want to uncover molecules involved in a cellular pathway. So that could be anything like antibodies, enzymes, receptors, or transporters. Secondly, we probably want to learn about the consequences of these interactions in this pathway. So that could be things like phosphorylation and dephosphorylation, acetylation, transport and translocation, and transcription regulation. And then finally, we might want to look for ways to control those pathways. So this is, usually the aim of a drug discovery project. So we might want to block a receptor with an analog ligand, to mutate or modify a ligand, and to indirectly modify effector molecules. So, just talking more generally about drug discovery, it is also becoming increasingly complex. So the targets are becoming more and more challenging and more explorative. So these could include things like membrane proteins, RNAs, protein complexes or aggregates, intrinsically disordered proteins, or what to date may have been considered undruggable targets. So, the range of targets that we're needing to explore and exploit for drug discovery is getting larger and more challenging. And this complexity means that it's also being addressed with increasingly innovative and diverse therapeutics. So that could include anything like biologics, immunotherapies, gene therapies, and a range of small molecules such as peptides, fragments, aptamers, and proteins. So there's now also a diverse range of different ligand classes that we might be looking at to address these drug discovery challenges. So today, as I said, I'm going to talk about two technologies, which are MST and TRIC, that can measure a range of diverse biomolecular interactions. And this could be throughout the drug discovery process, or it could indeed be in any project that seeks to understand cellular processes. So we have here the overall stages in a drug discovery project, starting with target identification and validation. And at this stage, MST and TRIC, so actually the two devices, which I'll come to briefly, that measure MST and TRIC are the Monolith and Dianthus. So at this early stage, we're using these technologies to investigate disease-related signaling pathways and protein interactions in vitro. So just sort of establishing those pathways and interactions in those pathways before moving on to, I guess, a larger drug discovery project. We then have hit identification. So this is where we would say, you know, we have our target and we would perform a single dose screen for this target across a wide range of ligands. So we throw a bunch of different potential ligands at it to see if they bind. And this is generally as a single dose, so just a yes/no binding response. Then if we have hits from that, we move on to hit validation. So again, we use these binding technologies to perform affinity screens. So we look at the affinity of different potential binders to the target. And then finally, we can also use it in lead optimization. So if we have some leads for potential ligands, we can use this to, you know, as they are developed, you know, say for higher affinity, we can continue measuring them with MST and TRIC. So we can use it throughout the drug discovery process and in a very early stage when we're still exploring a target and its interactions. So before moving on to the specific examples, I just want to firstly briefly introduce you to how MST and TRIC actually work. So just starting with the absolute basics of measuring binding affinity. So the goal when we're measuring affinity is to quantify the strength of an interaction between binding partners, so between our target and our ligand. And the resulting measurement that we get from these technologies is a dissociation constant, or the KD. And this is typically in the range of picomolar to millimolar. And this KD, it represents the likelihood that molecules interacting will break apart. So if we have a low number KD, so that's say in the picomolar or nanomolar range, this indicates a high affinity, so a strong binding. And if we have a high numbered KD, so say in the millimolar range, this indicates a weak affinity, so a weaker interaction. Now, what are MST and TRIC? Well, microscale thermophoresis, or MST, and TRIC, which is a temperature-related intensity change, they're related biophysical techniques that measure the strength of an interaction between two molecules by detecting variations in a fluorescence signal as a result of an IR-induced laser temperature change. So this is just a video of what that actually looks like in practical terms. So what we do is we have one molecule that's labeled, generally that's called our target, and we induce a temperature change with an IR laser, and we monitor the change in fluorescence of this fluorescently labeled target. So here we have fluorescently labeled DNA, and at the beginning of the video, it's sort of evenly distributed, and then I'll just play the video. We switch on an IR laser, which heats up a portion of this. So this is, you know, under a microscope, it heats up a portion of this solution, and you can see that there is a decrease in fluorescence as those spots are heated. So that's really essentially what we're measuring. We're measuring changes in fluorescence in response to heat. So the binding events then with MST and TRIC are detected by measuring changes in fluorescence intensity. So as I said, we label our target with a fluorescent probe, and we then monitor its change in fluorescence in response to an increase in temperature. And this is generally over a time scale of say 20 seconds. Now, in MST, there are two components that go into this overall change in fluorescence that we see. One is thermophoresis, and that's the directed movement of molecules within that temperature gradient. And the other is TRIC, the temperature-related intensity change. So these two components, so this is actually the response of the fluorophore itself to that change in temperature. So there's the fluorescent probe itself will undergo a fluorescence change in response to a change in temperature. So while MST measured with the Monolith, it takes in both of these components because it's measured in capillaries. So it includes the thermophoresis component. Whereas with the Dianthus, we're just measuring the TRIC component because this is measured in 384 well plates. So because it's measured in microwells, it's only the TRIC component of the signal that we're looking at. So that's the distinction, sorry, between the two. So we then, we see a difference in this fluorescence change of the labeled target, depending on whether it's bound to a ligand or not. And this is because ligand proximity to the fluorophore can affect that change in fluorescence, and also conformational change in response to binding can also affect this change in fluorescence. So really then what we're monitoring is the change, the change in the change of fluorescence between a target, a fluorescently labeled target that's in the unbound state versus being in the bound state. So finally, the way that we would actually derive a KD from this is that we measure this temperature-related fluorescence response for the target, with different concentrations of ligand. So say we may measure 16 different concentrations of ligand, and we look at then the difference in the relative fluorescence for each ligand concentration. So if we plot the relative fluorescence change against the ligand concentration, we can then get a curve to derive the KD for that interaction. So that's essentially how it works. And just to quickly introduce you to the two main technologies that we use to measure this, the Monolith is what you would use to measure MST. So some of the main advantages of the Monolith is that it uses very low sample consumption, so very low amount of sample. You can measure your KDs independent of the size and mass of the binding partner. So it doesn't matter if you have, a small molecule binding to a large protein, that's still fine. You measure it in solution, you can measure it in close to native conditions. That means you don't have to worry about immobilizing it on a chip. And it also means you can work with any buffer that keeps your protein stable. You can measure a broad range of affinities, so that could be anything from picomolar to millimolar, and do fast assay development. And you can measure many, many different types of sample. So, and many different types of interactions with the same device. Then if we move on to the Dianthus, so that's what measures TRIC. This is then if you want to scale up to a more screening approach, so a large scale screening. So this will analyze 384 data points in 30 minutes. So that could be up to 10,000 compounds per day. And you measure directly in a 384 well plate. There's also no fluidics, so there's not really any maintenance to be done or downtime with the device. And it measures, like the Monolith, a wide range of affinities and uses a small amount of your target sample and your ligands. So again, you can characterize in solution, so you don't need to immobilize, and explore lots of different types of targets and interactions with the same device. So they're very similar, just the format is a bit different for each, and I guess the throughput. And just finally, a quick comparison between some other common technologies that you would use to measure binding affinities. So SPR and ITC are two of the most commonly used. And this is just an interaction between, I think, a Cy5 labeled DNA and regulator MLC to show that with each of the technologies, we get a comparable, a similar KD measured for the interaction. However, in comparison to SPR and ITC, there's a smaller sample consumption for MST and faster time to get results. Unlike SPR, you also don't need to immobilize it on a chip. So, as I've said a few times now, we can, with MST and TRIC, we can address a diverse range of different challenging interactions. So whether that's part of a drug discovery program or not. So we can characterize things like proteins. So that could be anything like antibodies, membrane proteins, receptors, enzymes, and nanobodies. We can characterize binding with small molecules. That could be fragments, ions, nanoparticles, peptides, and carbohydrates. With nucleic acids, so DNAs, RNAs, or aptamers. We can look at virus particles and empty capsids. We can look at platelets and whole cells in some setups, and at vesicles like exosomes and liposomes. So we can address a wide range of different biomolecular interactions. Now, this example I wanted to show you first, it's mainly, it's designed to demonstrate the use of our histag specific dye with a range of different interactions, but I wanted to primarily use it just to kind of quickly give them one snapshot an idea of the versatility of MST and TRIC to measure a range of different interaction types. So in each of these interactions, the target was labeled with our his tag dye. And this is sort of an easy and gentle labeling procedure that will specifically label the his tag on your protein. So in the first interaction here, what we have is an enzyme that's been labeled with the his tag dye, and that's binding with an inhibitor. So you can see here a nice binding curve. We have, very small error bars here, and we get a KD of around 100 nanomolar. Then we have a similar example here, which is the P38 kinase with a small molecule inhibitor. Again, the P38 kinase has been labeled with the his tag dye, and we see then binding to the inhibitor. Again, KD of around 124 nanomolar. So to, protein small molecule interactions here. And then finally, we have a protein-protein interaction. And this has been done in two different ways. So we have the two proteins, the PUL53 and PUL50 interacting, and the PUL53 in this case is the target protein, and the other one is the ligand protein. So we basically just use target and ligand to distinguish what's the thing that's been labeled and what's the thing that's unlabeled that we're titrating. So here we've done it two ways. First, we have the purified target protein that's been labeled with the his tag, and then the other protein, the PUL50, has been titrated. So we see in the blue the binding between the two purified components with a KD of 1.2 micromolar. But then also the target protein, the PUL53, has been labeled directly in lysate. So with the his tag labeling, you can actually label directly in lysate if your his tagged protein is the only his tagged thing that's in there. So in this case, we have the PUL53 labeled in lysate, and again, purified PUL50 titrated. So we can see that we get a very comparable interaction for that as well. So 1.78 micromolar for that interaction. So we could do the interaction both ways, either in lysate or with the purified components. So that just gives you an overview of a few different types of interactions and how to use the his tag labeling, but I'm also going to go into a bit more detail about other types of interactions as well. So here we have a protein-nucleic acid interaction. So it's an example of how MST has been used to help unravel the binding mechanism of a CRISPR RNA to a Cas9-like nuclease. So in this case, the nuclease, the protein, was fluorescently labeled, and the RNA was titrated. But it's also common to do it the other way around, for example, by using a Cy5 labeled RNA and titrating an unlabeled protein. So you could do this experiment both ways around. So in this study, with the use of MST and in combination with other techniques, they revealed that this particular CRISPR RNA recognition mechanism, it leads to a conformational change of the nuclease. And they also used MST to show that the binding of the CRISPR RNA, it was actually sensitive to magnesium. So what we have in the blue curve is we have the binding of the RNA with the nuclease in the presence of magnesium with a KD of 2.86 nanomolar. Then EDTA was introduced to remove the magnesium. And what we have then is a decrease in the affinity. So we have an affinity of 154 nanomolar rather than around 2 nanomolar. So you can see the decrease in affinity in the absence of magnesium. So then, this overall, it opened the possibility for generating different mutants of this nuclease that have more efficiency and more specificity to their target, in order to ultimately improve gene editing tools like CRISPR. Now, in this example, we have MSTs been used to measure an interaction between DNA and a methyltransferase protein, and this was in order to see if it could be used to replace an electrophoretic mobility shift assay, or EMSA. So in this case, the DNA was labeled with Cy5, and the unlabeled protein was titrated. And this revealed an interaction between the protein and the DNA with an affinity, with a KD of 50 micromolar. So this just kind of illustrates quickly that, MST is as simple as an EMSA, but it's faster and it doesn't need to use radioactivity. And you also get a quantitative KD for the interaction. And finally, just a couple of examples with membrane proteins as well. So here we have a study on the structural basis of nitrate binding to a 12 transmembrane helix transporter, the NRT 1.1. So this is a protein coupled transporter, and it belongs to a family of membrane proteins that are responsible for nitrogen assimilation and nitrate transport in plants. And it switches between a low affinity state and a high affinity state, depending on, or in response to falling nitrate levels, and this is via a process of phosphorylation. So in order to better understand this mechanism, MST was first used to determine the effect of phosphorylation and point mutations on nitrate binding. So what we have here, firstly, is we have this transporter, which was expressed with a GFP fusion and purified in DDM detergent. So in this case, rather than labeling it with a dye, the GFP was used as the fluorescent probe for the MST experiment. So what we have first is the binding between the wild type transporter and nitrate, and you can see there's a KD of around 1 millimolar, that's in the red. Then with 1 point mutation, we can see that there's actually no difference to the binding of nitrate. However, with a different point mutation, we can see that there's no longer any nitrate binding. So it sort of helped to figure out which are the critical residues for the nitrate binding. So by combining MST and also crystallography, they revealed the key amino acid residues for the nitrate binding and subsequent transport. And it just helped to, I guess, further understand how the phosphorylation of the transporter allows it to switch between binding states. And it's also a nice example of, I guess, how sensitive MST is, because it allows you to measure the binding of a very small molecule or very small ligand to, a very large protein in solution, even when there's a very weak affinity between the two. And in this next example, it uses both NanoDSF, which is a technique for measuring protein unfolding, and MST to help understand the structural basis of a drug and peptide binding and transport. So the human PEP-T1 transporter, it's an important pharmacological target due to its role in uptake and transport, in drug uptake and transport. And so in this study, the researchers, they've used a bacterial homologue, the DTPA, and this was to, help to better understand drug binding and transport of the human transporter, of the human protein. So they firstly used NanoDSF. So this measures the temperature of unfolding of a protein. And this was used to screen for nanobodies that stabilized the protein in order for, to aid for crystallization and structure determination. But they also used it to do what's called a thermal shift assay. So this, looks at the difference in melting temperature of that protein in the presence of different potential ligands. So what they've done here is they've tested it in the presence of a variety of different tripeptides, dipeptides, and drugs, and looked at whether or not they've stabilized the unfolding of the protein. So they've increased the melting temperature of the protein. And the inference here is that something that increases the melting temperature of a protein or stabilizes it is doing so by binding with the protein. So they then went on to quantify this binding with MST. So in this case, it was label-free MST, and it was used to quantify, a variety of those different peptides and drugs to the DTPA, and also to try and find out which residues were involved in the binding. So it's quite a large paper where they've done binding on a lot of different mutants with a lot of different ligands, but this is only just showing one tiny bit of that. So in this case, it was label-free MST. So it was the purified DPTA, transmembrane transporter in DDM. And so rather than attach a fluorophore, they've used the intrinsic tryptophan fluorescence of the protein, which can be used with label-free MST. So here we just have the binding curves, the MST binding curves for the DTPA alone, or in complex with a nanobody that was used to stabilize it for crystallography. And this is, we can see it's binding to one of the tripeptides, we can see that we have a similar binding affinity regardless of whether the nanobody is there or not. So just one example of, of the binding curve with one of the potential ligands. And then finally, they confirmed with crystallography, how the various mutants, responded and basically revealed the binding site of one of the prodrugs on the DTPA. So that's just, I guess, a brief overview of some of the different types of interactions that you can measure with MST and TRIC. So I just wanted to pause for a moment before moving on to some screening examples to just point out that we have many more, I guess, in-depth webinars and stuff on our website. So you can look at topics like membrane proteins or plant research, gene therapy, virology, and fragment screening, compound screening in more detail on our website with any of these webinars. So now we've seen, some examples of different types of interactions that we can measure with MST and TRIC. I just want to show a couple of examples of how this could be scaled up for hit and affinity screening for a drug discovery project. Okay. So the examples that we've seen so far would most likely fit into this first category, which would be target identification and validation in a drug discovery project. So this is, where we're investigating disease-related pathways and identifying the targets and pathways that, we would want some kind of therapeutic intervention for. So once the target has been validated, we then move on to the next stage, which is hit identification and validation. And this is in order to find, more potential ligands that will bind to the target. And these could be anything like fragments and small molecule compounds, nucleic acids, peptides, other proteins and more. So the first example that I want to show you is a fragment screen against the target G9A. So G9A is a methyltransferase and is involved in epigenetic gene regulation processes. It's also been identified as a potential target in breast and lung cancer. So this project was actually carried out in collaboration with Imperial College and the Francis Crick Institute. So the group there had been using MST to measure the ligand interactions with G9A, on a smaller scale. But we then worked with them to basically scale this up to do a larger fragment screen with the Dianthus. So for this screening, we lysine labeled the G9A with our TRIC optimized dye, and we screened a library of almost 2,500 fragments against it. And this covered a broad, range of chemical space and mass and complexity. So the first step is to establish your positive control interaction. So here we have the G9A with a positive control ligand, a known binder, which is SAM. And we have first the single dose test. So here we are just testing, we have basically the target just with reference, which you use usually DMSO. So if your ligand is in DMSO, you use DMSO as a negative control. So we have our target with DMSO, and then we have our target with the positive control ligand. And we want to see that there's a difference between the TRIC response of the target only and the target with its ligand. So we have a nice difference here. And we also then do an affinity measurement. So we have, a serial dilution of the ligand to actually measure the KD of the interaction. So the KD in this case is 13 micromolar. So first we establish these two, that we see, we have a nice signal amplitude, nice signal to noise that we actually see binding with our positive control ligand before we move on to do a full screen. What we can also do is a buffer screen just to make sure that we have, the best conditions for this assay. So here we did a buffer screen. Again, we have our labeled target with its positive control ligand, and we're basically just testing yes/no binding in a range of different buffers. So we have a specific buffer screen to do this. which has 96 different buffer conditions. And on the Dianthus, it will give you this heat map of which are the best conditions for binding. So which conditions give you the best signal quality. And by doing this, we actually identified that there was a slightly better condition than what they had been using. So the buffer that they already had was very good, but we did find one condition that actually improved the signal quality. So we then moved forward with that buffer for the full screen. So the next step was to do the single dose screen with the 2,490 fragments. So what we see here is, so overall to screen these, almost 2,500 fragments in duplicate, this took 6.5 hours of measurement time, and all of the ligands, they were tested at a final concentration of 1 millimolar and a DMSO concentration of 1%. So again, as a positive control, we use the SAM, and you can see this in the yellow. So throughout the screen, we make sure that there are positive controls, scattered throughout to make sure that, that the target is still, it's still responding to its ligand, it's still functioning correctly, it's still stable, and we want to make sure that it's basically stable over the whole measurement time so that this positive control interaction looks the same at the end as it did at the beginning. And also, to reference things throughout the experiment. We also, throughout the time, scatter reference measurements, which is just in the DMSO. So then we see, some of these, so hits would be in the blue. We see the points that are hits. These are just relative to the reference. So we have all of the references here. Then we would have non-binders here in the middle as well. And they would have to deviate. You can set how much they deviate, but, say we might set a criteria of, it has to have a response that's at least 3 standard deviations away from the reference to be considered a hit. So then we have, the ligands out here that could be hits or potential hits. We also have other categories like aggregators or things that show autofluorescence. I mean, this is all, we can go into a lot of detail, but we won't right now. But just to say that it will perform the screen and then it will automatically categorize all of those fragments or compounds or whatever it is that you've tested. So in this case, we got 89 hits, which we then took on to do an additional affinity screen. So with any of those that were hits, we tested them with a full dose response to see whether we could actually measure a KD for them. And so that screen then took an additional 1.5 hours. So to screen, to do the full binding curves for each of the 89 hits was just another 1.5 hours. So then just to summarize for this screen, so firstly it was a single dose screen with the, say, 2,500 fragments, and then a follow-up affinity screen with the 89 hits from that single dose screen. So overall, it was 2,500 fragments in duplicate in less than 8 hours. That's including the affinity screen as well. It used less than 20 micrograms of the target protein, and the KDs were a range of different affinities. So that was in the range of high micromolar to millimolar range, and that's pretty much what you would expect for a fragment library. Generally, you would expect pretty low affinities at the fragment stage. And we had a good agreement for the positive control ligand KD based on ITC data as well. So the positive control interaction, it agreed well with ITC. So this basically is just summarizing the same thing again, that, we measured the whole 2,500 compounds in approximately 8 hours using a small amount of sample, and that was covering a broad range of different molecular complexity of the fragments. So the final example here is just a compound screen against the P38 mitogen activated protein kinase, and this is a stress activated serine threonine kinase. So this kinase has a role in regulation of various cellular pathways, such as apoptosis, inflammation, cell growth, and proliferation. And it's also been identified as a potential target in a range of diverse diseases. This is including breast and lung cancer. So again, the first step is in setting up the assay is to label the target and test its interaction with its positive control ligand. So actually what we've done first here is to label the target and then test its thermal stability, so test the unfolding in the absence and presence of its ligand. So we see here that it's still stable after labeling. And then when we introduce its ligand, we see there's a thermal shift. It's further stabilized, inferring that it's still active, it's still binding its ligand. And then so it's looking like a good quality sample to proceed with the screening. So firstly, here is the titration of the positive control ligand. So again, we do a serial dilution dose response and measure the KD. So there's a KD of 100 nanomolar for this positive control. And, you know, it's got a nice signal amplitude, nice signal to noise. So we know that it's fine to proceed with the full single dose screen. So here we have a screen against 170 compounds. So, you know, after we'd established that the assay conditions were good with our positive control, we proceeded to do this single dose screen. And this is a commercially available kinase library with 170 different ligands. So each ligand, this is performed in duplicate measurements, including several different reference samples, in this case DMSO, and several different positive controls measured throughout the plate, throughout the assay. So again, you know, we see the scatter plot of our reference area, we see our positive control measurements in yellow. So overall, we had 57 hits from this screen. And then some other thing like, you know, compounds that cause aggregation or cause autofluorescence, some that are inconclusive that you could look in further. There are different classes of inconclusive, which I won't go into more detail now. But overall, so these 170 compounds in duplicate, we could fit that onto one 384 well plate. So the measurement time was 30 minutes for this experiment. So just to summarize, that's 170 compounds tested in 30 minutes, and TRIC uncovered 21 additional hits compared to a traditional thermal shift assay, and it only missed 4 hits. So there were a very low number of false negatives and a high number of unique hits for the technology. So, you know, now you've seen how Monolith and Dianthus, they can be used to scale up a drug discovery project and to perform hit identification and validation. You can also then continue to use them both throughout the lead optimization phase to continue measuring affinities for potential ligands identified at the earlier stage. So hopefully, you know, you've got a good overview of how you can use both MST and TRIC to study a broad range of different biomolecular interactions throughout the whole drug discovery process. And even, you know, I guess disregarding drug discovery process altogether, just in any project where you want to investigate cellular pathways or you want to further understand cellular signaling. So anywhere where you would be interested in looking at interactions. So again, before I finish, I just want to highlight that you can find a lot of additional resources on our website if you'd like to take a deeper dive into any of these topics. So there are many different webinars there. There are also some spotlight pages on various topics like gene therapy or membrane proteins or virology, plant science. So yeah, have a look if you're interested in, you know, taking a deeper look at any of those topics. So finally, that brings us to the end of today's seminar. So I'd like to thank you for your attention, and thanks to Filippo for inviting me to speak. And I'm happy to take any questions now that you have about what we've covered today.
Thank you very much. That was great. Thank you for giving this talk. I actually got a few questions. I'll start and I'll ask two. I don't want to be the only one asking questions. So you mentioned at the beginning the cell lysate, that you were labeling directly this target, the cell lysate. How do you treat the sample? So, because that was the monolith. So that means that are you actually putting your sample in the capillaries directly?
Yeah. So I mean, generally you would, I guess there are different ways that you would potentially prepare the cell lysate, but I guess get it to a fairly homogeneous form, you know, and say solubilize, like if you were doing something with a membrane protein, you know, you'd want to have detergent in there as well to solubilize the membranes a bit. But yeah, basically, we have some protocols on our website for how various people have done it, but yeah, preparing your lysate, maybe doing some, I guess, enrichment steps, so not quite purification, but you know, you kind of get the debris out or whatever. Yeah, and then there are a few more pre-steps to labeling, you know, to I guess try and establish the concentration of your target in that lysate, to find the right amount of dye to use. But then essentially at the end of the day, it is just mixing the dye into that lysate in a ratio that you know all of the dye will be bound to a target. And then you can use that directly in the capillaries. So for those experiments in lysate, you should also have a proper negative control as well.
When you got the GFP, because I remember, I remember from the very beginning I started working with MST, one of the first things I was told is try always to use the red channel and avoid. How does the GFP works from there?
So the GFP is actually in the blue channel. So we generally recommend, so with MST, you know, your two most common filter set combinations are red and blue.
Yeah.
But always, I think whatever you do, always have the red, because with our dyes, we optimize them all and they're all made for the red detector. You know, I guess they're the most sensitive, so we have least interference in the red range, but we also appreciate that, you know, a lot of people use things like GFP, which is measured in the blue channel. So that is an option as well. So in which case, yeah, you're measuring with the blue fluorescence detector rather than the red. Yes, it is a bit less sensitive. So you might need higher concentrations of the GFP labeled target. I think also it's a little bit more prone to photo bleaching depending on the type of GFP you have. But I mean, also having said that, people are very successful using GFP as well. But I guess the red channel allows you to drop things to a much lower concentration. Like if you've actually covalently labeled with a dye, you could be using your target at, you know, low nanomolar or picomolar concentrations.
Yeah. Last question, then I'll leave it. When we start doing like drug screening, finding out that there is a good drug that binds the protein is a good starting point. But most of the time, we are, especially when we start grant application, we get asked, okay, have you shown that there is biological activity? For some of these compounds where you got KD in the decent micromolar range, so the 10 to 20, 30, like good direction, but not quite good yet to go in cell lines, you've got to show some sort of at least some biological activity, so level of competition with the natural substrate or things like that. So can we do competition assays within the TRIA, the Dianthus?
Yes, definitely. So I didn't actually show it today, but yes, you can set up a competition assay. You can set up, you know, ternary interactions very easily. So I mean, it would be a matter of first, you know, say establishing the interaction between, you know, say your natural ligand and your target.
Yeah.
And then you would, you know, add the natural ligand at a fixed concentrate, a saturating concentration, and then you can titrate in the other compound and see then what the EC50 is essentially.
So it doesn't matter how many things you put inside the assay?
No. I mean, it doesn't matter how many things you put in. I guess then it more matters is, can you deconvolute? Can you understand what's actually happening? But definitely, you know, competition assays are very common. Say if you have a tracer ligand. So I think one of our most common examples is having fluorescently labeled ATP and competing that off. So yeah, if you look on the website, especially in the Explorer community, you'll find examples of competition assays. We also have an example that I didn't show today, but in the Dianthus webinar, it's called High Hanging Fruits on our website. It also goes into how a peptide library was screened as a competition assay on the Dianthus. So yeah, it's very doable, just a slightly different assay setup.
Okay, is there any question from the audience? Anyone wants to ask a question? Otherwise, I can go on. Mike.
Yeah, very nice talk. Thank you. Very interesting technique. I've never heard of it before. What happens when you have the interesting case of some sort of fairly allosteric system where you are binding a ligand and then, then of course the shape of the binding curve is different. So I guess you pick this up, but I don't know if you report back a KD or because now there are two ligands perhaps involved, and then the KD won't be right because you won't have the right units and so on. You have systems to account for that, do you?
So are you saying if you've put in ligands, then one's an allosteric?
Well, the protein's allosteric and you add something, and then that's binding not independently of other ligand or in other ligand. So that would change, it'd be a binding curve. You show some binding curves that standard across 2 decades. And then, but sometimes you have something which is very tighter because it's allosteric. I wonder if you have ways to handle that.
So, I mean, at the end of the day, so if we've got a binary system, so if we've just got 2 components in there, we'll just be reporting the KD.
Yeah.
But then if you have, multiple components in.
No, it's only 2 components, but but they bind allosterically. So,
that
so
we can't necessarily.
It's the binding of the second, 2 binding.
Yeah, we can't necessarily tell the difference. We just get the overall KD.
Right, you get the midpoint, yeah.
Yeah, in terms of, I guess, looking into mechanisms of binding, you could start, I guess, looking for patterns, different types of ligands in the direction of the curve.
I guess you would actually see, your system, as I understand it from what you said, you would see whether something's allosteric by the shape of the curve, actually. You wouldn't have to do very much, but you have to do something different with the analysis, probably.
Yeah, I mean, and then you can, for example, if you have something that, shows cooperativity, you can fit it differently. So then you could fit it with the Hill model instead, and then then we get an EC50 rather than a KD. So
so you can do that. Okay.
Yes, we can do that. So then it is. It is very dependent on that exact situation, which is why we report an EC50 rather than a KD.
Yeah, I get you. Okay, thank you.
And probably linked to that, I remember back in the days, I actually tried to do it once, but never managed. Stoichiometry. I remember it's possible. It's not that straightforward though, because that's the main thing that you get from ITC, but
yes. So it is possible. Actually, I think my next slide, if it's still up.
I swear, this was not prepared.
Was to say that yes, so you can measure more than binary interactions. Stoichiometry is possible, but it does require a slightly different setup in that you have to do a few steps of the experiment at different concentrations of your target. So you essentially have to have a saturating concentration of target as well to then calculate that sort of kink to get the stoichiometry. But it's possible, and we have protocol in our Explorer community to walk you through how you would set up that experiment.
Okay, any more questions? No? There's no more question. Mike. Mike, you're muted. There you go.
Another one. I see you got thermodynamic things down there. So you do that by changing the temperature of the actual. So your whole thing is temperature control. So although you change the temperature, there is a basic set level that you change that for?
Yes. So it actually depends whether it's the Monolith or the Dianthus. So the Monolith, you can temperature control from 22 to 40.
Right.
So you could do it at different temperatures. The Dianthus, because it's been designed with screening in mind, it's a bit more limited in temperature. So I think it's 20 to 25.
I was wondering how much does the laser heat the solution?
So it does depend on the measurement times, but I mean, so say a standard measurement, it would be no more than 10 degrees increase by the end of the measurement. But there are different time points at which, you know, we can, I guess, guarantee that it will be no more than 5 degrees or no more than 5 degrees. So yeah.
I was wondering if they ever, you know, caused a problem, because your KD is also temperature dependent. So it'll be different at the beginning and to the end of the experiment. So you're measuring some sort of average, aren't you, really?
So it can be that the KD does, I guess, change. So we aim for the least, I guess, put into the system. So we aim to change the system as little as possible. So we have what's called different, I haven't gone into this, but we have different powers that we can use of the IR laser, which give different rates. So actually with some experiments, you could have only a very small temperature increase over the whole 20 seconds. Or you could even, in most cases, be able to see an interaction at 2 seconds where you've hardly had any heat change. There would be some situations where some interactions do change depending on the time. So you might get a different KD depending on which time point you sample. But that's not all interactions. So what that tells you is that the interaction is being, or like perhaps the target is being destabilized by heat. But these are things that we, I guess, you know, we kind of flag up as you should look at this, you know, time points you should use to guarantee that you have like the least heat change. And, you know, if you see phenomenon, you know, this might be the reason, and this is what
Anyway, that's good. Thank you. It's very fascinating. Thank you.
No worries.
Okay. Any other question? I'm conscious about the time. Okay. I don't see any question in the chat. Okay, in this case, I'm gonna thank you, Claire, again for the great talk, and and thank you for explaining the technique so clearly. Thanks a lot.
Okay.
Thank you. Thank you.
You're welcome. Thank you.
Bye.
Okay. Bye, everyone.